Thank you, Kumar. Hi, Jonathan Spector here. Hi again, everyone. We met earlier in the day, and what a sensational day of science it has been. We're so grateful to be able to end on such a high note, and it is my pleasure to introduce Dr. Christine Kreuder Johnson, who is the Professor of Epidemiology and Ecosystem Health and Director of the EpiCenter for Disease Dynamics at the One Health Institute, both at UC Davis in California. Professor Johnson earned her doctorate in epidemiology from UC Davis and a veterinary MD from the University of Pennsylvania. She is a pioneer in what must be considered to be one of the most relevant fields of study at this moment in history, transdisciplinary research that pulls back the curtain on real-world dynamics at the intersection of the environment, animals, and human health to reveal instigators and enablers of disease emergence, spillover, and spread. Among the viral pathogens in her line of sight, Ebola, Arbo, and of course, Corona. And because this work is foundational to informing science-based decision-making and public policy, there's no question that it directly influences us all. We could not imagine a more impactful way to round out this day of COVID science. To the audience, please remember to put your questions in the chat at any point during the talk. Thank you enormously for being here, Chris, and the floor is yours.
Thank you, Jonathan, for that very kind introduction. It's a pleasure to join the Symposium on the Science of Therapeutics. I'm so grateful to you all for advancing the research that you're doing for the current pandemic. My field, as Jonathan said, it's about as far removed from you all as possible while still being in the realm of medicine, I guess. I'm a veterinarian and an epidemiologist, as he said, and like many with my training, I find myself starting a lot of work on the epidemiology of emerging infectious diseases, specifically how diseases emerge from animals to cause pandemics. And I should mention before I get started that my views are of course my own, and I'm not speaking on behalf of Novartis. And normally my work is not at all that relevant and certainly not controversial in any way. But I'm sure you've noticed in the last couple of weeks, especially the origins of the pandemic has been a source of a very heated debate. And as one might imagine, given the toll that SARS-CoV-2 has taken on humanity over the past 18 months, understandably, people want to know what caused this. And honestly, I thought about changing the plan for my talk, but then I thought, it's always best to stick with what we know best, right? And spoiler alert, I don't have any special insight into what caused the pandemic. And that's because these things take time. My work has investigated the circumstances involved in emerging pandemic threats for the past couple of decades. And all of my work has been done through the great work done by many brilliant scientists. And I look at how these viruses have come to spill over from animals to infect people in the hopes that that this can inform pandemic preparedness. And so I'm very excited to talk to you all about the breadth of this work and what we know and in many cases how much we do not know. So I think you can all see that slide. That this slide is a slide I've just had forever. For presentations, but most importantly to teach students that I train at the veterinary school and in the programs of epidemiology. And I talk about emerging infectious diseases and sort of the circumstances around their emergence, like I said, through work done by many, many others. And I think that's, once you start to look at all these together, you start to understand some clear patterns. And so let's start with Zika. Zika virus, of course, emerged in Uganda, the reservoir primates. There was a public health emergency declared recently, as we're all aware, following widespread transmission in South America. And then there was Bolivian hemorrhagic fever. This is caused by Machupa virus. It's an arenavirus transmitted from field mice in Bolivia. It's caused sporadic outbreak with very limited human-to-human transmission. Then, of course, we have Ebola virus, which was first recognized in Central Africa in 1976. It's caused repeated outbreaks since then until it became very much on everyone's radar with a catastrophic outbreak in 2014 in West Africa, and has sadly continued to spill over several times since then, mostly in Central Africa, most recently again in West Africa. Bats are the suspected reservoir. Sometimes people get infected through other animals. There's a lot we don't know about that. And of course, there's hantavirus pulmonary syndrome, emerged in 1993 in the Four Corners area. That's in the US, so we've got one of our own. Changes due to rainfall, creating drought and then a surplus of rain, created the initial pandemic, and the deer mouse is the reservoir that infects people through transmission. And then we have Nipah virus, which was first recognized in 1998 in Bangladesh. It's a paramyxovirus, so another virus we need to keep tabs on. And it's got a host of Pteropus bats. They're so big, they're called flying foxes. And that virus has similarly, actually at first emerged in Malaysia with a pig outbreak, similarly caused by Pteropus, and then has emerged in Bangladesh repeatedly over the last couple decades since it was first recognized. And then, of course, we have SARS, the first SARS in 2003, which emerged in a market setting also in China, and has a bat reservoir, although the circumstances around the transmission and the involvement of civet cats is still being pieced together very much. And then MERS came on scene in 2012 through camel herders who got sick. And we know that camels are an important domestic species reservoir for MERS, but the viruses have actually more than likely have a bat reservoir as well. And so what you can see that's in common with all of these is that there's an animal reservoir, importantly a domestic animal reservoir, occasionally on the way to transmission to human, but always in all of these cases a wild animal host. And what should be noted is that the specific animal species and the human activities involved in facilitating disease emergence have come to light many times, years and years, if not decades after the initial event. And so that, I think, just highlights some of the complexity and sort of the research endeavors that need to come to play around these issues. And that's because there are literally thousands of viruses in animals. Coronaviruses alone, there's probably hundreds and hundreds in animal species. And there's only a very tiny fraction of these that could be pre-adapted to be zoonotic or infect humans, or be evolving to be zoonotic. And so this gives us a really broad array of usual suspects, if you will, to consider. And we think that the evolution of a pandemic threat sort of goes along these lines. So we have this, of course, this evolution or adaptation of pathogens, and really important to keep in mind that this has to do with viruses that are emerging on what we can only describe as a very rapidly changing landscape on which we share this planet. And occasionally these viruses will start to transmit between animal species. And once they start doing that, we consider that a bit of a red flag, because these viruses can then be on their way to spill over into humans. This cross-species disease transmission is really common for viruses that end up being zoonotic in the work that we've done. And occasionally, around 40% of these viruses end up becoming human-to-human transmitted. So most spiller events they're dead end. The human needs to contact the animal for there to be transmission. But on occasion, these viruses adapt and become human-to-human transmissible. The problem is, for novel viruses that, of course, started in the animal reservoir, humans have no prior immunity, and the conditions are set for an epidemic to take off. And with our globalized travel, it's not lost on anyone that for widespread viruses that can transmit this way, there's the high potential for a public health emergency of international concern once these viruses start crossing borders. My work has largely focused on viruses instead of other pandemic threats that might be caused by other pathogens like bacteria. And our viruses are really unique. We feel like they're highly adaptable, so they're much more happy to cross the species barrier and go unnoticed for a long time. So we have this sort of scenario that we expect to happen when there's an emergence of a pandemic threat. So we expect that there's outbreaks in animals that precede outbreaks in humans. And I would argue that unless the cases are very unique and there's an unusual cluster of disease with readily accessible healthcare, that these tend to go unnoticed. And then there might continue to be outbreaks in animals usually with one of these usual suspects, as you saw from the previous slide, bats or primates or rodents. And then eventually we get an outbreak of enough of a size with an enough unusual cluster of diseases, clinically recognized diseases. I would argue oftentimes these individuals end up in a healthcare setting, and a doctor or a nurse or someone in that setting gets infected, and that is when the disease is recognized. And for the most part, I would suggest that this entire picture is still very much shrouded in mystery, as Jonathan said, and we know almost nothing about these circumstances. And I think we could just take human coronaviruses for an example. These are an important pathogen. We've had numerous coronaviruses emerge that were a good warning of a potential pandemic, starting with SARS-CoV-2 in 2002, which caused severe disease, and then again with MERS in 2012. Yet there's a number of coronaviruses that circulate in humans, and we know they have animal origins, but we have so many questions, major question marks. All of these are suspected pathways. None of these have been truly confirmed. And of course, we have OC43 that emerged back in the 60s together with 229E that really has gone largely under the radar, causes the common cold, but has zoonotic origins. And then what I find really fascinating is two more coronaviruses emerged since SARS, or at least they were recognized after. NL63 in 2004 and HKU1 in 2005, also likely zoonotic transmission events, just didn't cause major disease. So you can see the questions that still surround a lot of this, and most of these have gone on to be widespread in the human population. And of course, depending on their pathogenicity, that is the question at hand. And I think we're really interested in knowing what leads to the emergence of a pandemic threat, of course. But I think when we're asking the question, how did these people get infected, from which animal source, we're really missing the bigger picture. For the most part, we want to think about emerging events as just like we were all taught in medical school or veterinary school alike, how these mechanisms of disease are largely dependent on the pathogen and the host, of course, but the environment. And I was just at a talk that showed basically this whole triangle being the environment, and the host and pathogen teetering on the very top here, with the environment swaying it one way or another in favor of the host or the pathogen. And I have to say, I really like that idea of the epidemiologic triad much better than this one, because it really highlights the huge role that the environment plays in disease emergence. Certainly, animal-human contact is important. And so our work has looked at the direct and indirect contact for wildlife that resulted in disease spillover for all of these different viruses. So these are the virus families that tend to have zoonotic viruses in them. And so we looked at all zoonotic viruses, and out of about 150 zoonotic viruses, actually it was a little bit less than 100 had any details about the circumstances for transmission from animal to human. And I've listed them here. The vector-borne ones are, of course, a little more easy to recognize once you find it in a vaccine. You're like, oh, that's it. That's how that one goes. But a lot of these are circumstantial evidence that was reported in the literature highlighting how people have gotten infected around their dwellings or in agricultural settings. And I've got sort of direct contact and indirect contact separated here in this network. There's infection from laboratory settings. And then there's a lot of direct contact that researchers have, that people have in zoo and sanctuaries, that hunters occupationally have exposure to wildlife that results in transmission. And so all of these red dots, if you will, are different viruses that have transmitted this way from animal to humans, just highlighting these high-risk interfaces that are really important considerations when we start to think about disease control. And so additional work that we did looked at, for all the different species on the planet, how many zoonotic viruses do these different species have? And you can see why rodents and bats tend to feature pretty commonly in emerging infectious disease events. That's because there's so many of them. So this here shows the percent of mammalian richness that is made up of rodents and bats. And there's just over 5,000 species of mammals on the planet. with us, 5,500 or so. There's another 10,000 birds, but we're just going to look at mammals for a minute. And you can see that a big proportion of them are actually rodents and bats. So it makes sense that these guys here are going to have a lot of diversity in terms of the different viruses that they might have, which just opportunistically, a few of those might spill over into people. Now, the case is really different with domesticated animals. Domesticated animals, of course, are really populating the planet, only outpaced by humans. This is sort of their global abundance. And what we found is that the number of viruses in each of these different domesticated species really closely aligned with how common these species are on the planet, suggesting that the more common species, the more viruses they've shared with people. And I think it's not lost on any of us that the conditions in which a lot of these animals are kept are really ripe for how these diseases can be transmitted. To start with that cross-species disease transmission, you can just imagine this being a little influenza incubator here, that then enables transmission to humans that are, of course, very nearby. And so our work really wanted to certainly highlight, of course, the very important role that wildlife play, but also how fundamental domesticated species are in disease transmission. So these are all domesticated species here in the center of this network. And then we've got all the other wildlife species that are linked together by a zoonotic virus. So among all the viruses that have been recognized to transmit to humans, domesticated species played a huge role in getting those viruses from some wild animal into the human population. And when you rank them as far as the species with the most viruses, the domesticated, the mammals, they rank at the top. And then just down here, coming in sort of the last couple places in the top 10 are, of course, rodents. Two really, really common rodents, again, super populated all over the planet. So no surprise there. A lot of my work just really points to what we already should know and understand based on the prevalence of these things. But we were really interested in taking it one step further. So what we wanted to look at was, for all the species that have shared viruses with people, what are some of their common characteristics? Like, what do these species have in common? Is there anything that we can understand from the different species that would dictate whether or not they're going to be more likely to share with humans? And as you can see, there we are with domesticated species. This is a model that we did that predicts the number of zoonotic viruses in that species based on what's been recognized in the literature through history. So domesticated species were over 8 times more likely, or had about 8 times more viruses in each species that had been shared with humans. And then following domesticated species, we noticed, similar to what we were alluding to on the previous slide, that the more common species shared more viruses with people. So animals that have been ranked and categorized by the IUCN, which is the International Union for Conservation of Nature, those species that are of least concern and increasing on the planet, meaning they're wildly abundant, they're maybe even what we call anthropophilic, they're mammals that have accommodated habitat change, different landscapes. They're able to adjust to human conditions, sometimes even dependent on human conditions. You can think of some of those field mice that require sort of the field crop habitat, or the house mouse, which the name implies likes to live in the house. These species that are increasing, and they've shared more viruses with people over time than species that are decreasing. And the trend is really quite linear. There are mammalian species that are near threatened, vulnerable, endangered, and even critically endangered, where we're talking very few animals here. And these had a much less likelihood of sharing anything with humans, had very few zoonotic viruses. And so then we ask, well, what is it about the species that are in these different states of abundance? Well, again, we need to control for the different taxonomic groups. And there's bats. Again, bats have after we adjust for that whole effect that I was talking about the whole bat group having a lot of different species, after we adjust for that, we learn that bats actually still share more viruses with people, even following that adjustment, as do primates. And primates, of course, they're very related to us. We share a lot of viruses back and forth between us. We give them viruses, they've given us viruses. It goes back and forth that way. But then when we get down to what I was most interested in, which is the environmental change aspect, we're looking at some factors here in the way species are being caused to become threatened. So species, mammalian wild species that have been in decline because of loss of habitat or loss of habitat quality, these species in particular have shared more viruses with people than species that were listed for other reasons. And similarly, populations that have been in reduction, so species losses, species being lost over time in terms of abundance, because of exploitation. So thinking about hunting and other things, these also shared more viruses with people. And similarly, species that have had reductions directly observed. So these are species that we know a lot about. These are species that are heavily monitored. There's big plans to monitor whether they're going to recover. And as we saw from a previous graph, some of those activities can increase disease transmission as well. They tend to be in close proximity to people. But let me be clear, the majority of species that are threatened on the planet are threatened because of declines in habitat quality and because their population has been reduced because of exploitation. These are major drivers of population decline, and it also turns out these are important factors related to whether or not they are likely to share viruses with us. And so just thinking about this on a broader scale, certainly we have species that are in global decline very commonly because of exploitation and habitat loss. And what we can expect then is that these activities that actually have caused the species to become less common are also activities that increase the risk of spillover. So exploitation of wildlife, typically done through hunting or putting live animals in the wildlife trade. This has been, I think, heavily conveyed over the last year and a half that this is a pretty risky activity. In many cases, we consider this sort of the perfect epidemiologic setting for spillover. The animals are often brought together in cages. They're moved over long distances. They're housed in cages with different species, sometimes with domestic species running around as well. And just like anything, and this is something we think about a ton in the veterinary field, if you're pulled up and moved to a new location and kept in dense conditions with other animals, that increases the amount of virus circulating in that little closed system. Animals are much more likely to get sick in those conditions. In cattle, we actually call it shipping fever because cattle will break with a disease that they didn't apparently have on the farm, but once you put them together in a truck, all of a sudden they've got shipping fever. And we suspect that the same exact thing is happening with wild animals that are transported in these settings. And then you can imagine many of these are being moved from nodes in a more rural wildlife trade setting into urban settings where there's just much more potential for human contact. So we start to look at the wildlife trade like a pretty dangerous probability game where at some point viruses that were in that species in the less disturbed habitat now have a much more open road into infecting people than they might have before. So that's something that certainly this work points towards. And then the other really important thing is to think about habitat. And again, we want to consider the environment and how that is influencing disease transmission if we're ever going to get at risk mitigation. And so the declines in habitat that were reported for wild mammals, for ones that were in decline or threatened with extinction, these were largely due to deforestation, development, conversion to cropland. And these are things that we suspect, big knowledge gaps here, let's be clear, increased disease distribution. We know that these things increase animal-human interactions, but we hypothesize that they play a role in disease dynamics. And I'll get back to that in 1 minute. But thinking about environmental change, we just have to acknowledge, of course, the major forces of global change that have been on this planet, because our whole goal is to understand why pandemics are emerging, and most importantly, why are they emerging at a faster rate? And we know human population growth has been exponential in recent history. I love this figure because it really puts things in context, going way back BC, and you can just really see how dynamic the human population growth has been. And this has also brought about major landscape change. So as we've accommodated the planet for our own populations, we've urbanized, we've industrialized, we've incurred major gains in agricultural development, and a lot of that has been mostly just really over the past few centuries. And of course, we've got movement and migration that never really happened at the scale that it has in the last 50 years, certainly. And I love this figure. This is another way to think about forces of global change. We don't just have the population increase, which is this trajectory down here. We also have these big drivers of change. So increase in affluence and increase in technology are major ways that we change our habitat, the habitat of animals, and that way we bring about potential for emerging infectious disease risk. So these are things that we need to take into account. We need to understand in terms of technology with motorized tractors and things that can deforest at a much quicker rate than used to be. Things that can make cropland much more abundant in a way that wasn't possible before motorized equipment. This is the type of technology where we start to really understand the power at which humans have been able to bring about change on the planet. And I think that's important to recognize when we're trying to think about how to interpret recent trends. So the way we think about some of this in terms of the environment is from an ecosystem health perspective. And that's how I've been kind of classically trained as an epidemiologist in ecosystem health. That's actually my professor title. And we want to recognize that there has been rather unprecedented global change. We have changed ecosystems through new technology to meet growing needs that the human population has had. Of course, for food, for fresh water, with dams, we've changed how fresh water is moved across the planet. We of course need timber, fiber, fuel, and other things, and those demands have changed the landscape. That's pretty obvious. And of course, that's brought about a ton of major gains in human well-being, really important things that we needed that brought about even greater population growth, but also economic development. But in this particular this figure and what I'm quoting down here, the Millennium Ecosystem Assessment, which was done by over 1,000 experts in 95 different countries at the turn of the century, back in 2005, they classified how many of these changes have brought about not just a change, but a degradation in ecosystem services. So fundamental things that we get from the ecosystem, like fish, like timber. So they've noticed that these activities have not necessarily been sustainable, and that some of those services, like fisheries, like forests, are declining to an unsustainable level. And this is a great resource, and I beg you all to check it out, just to see how their view of sort of where we stand and how these systems tally has been really interesting. But most importantly, I think from our perspective, is this change has brought about what we would consider a nonlinear or abrupt change in ecological processes. And I would argue that emerging infectious diseases is just one of those processes. You can think about these abrupt changes being from, we always had this fishery, the fishery existed for centuries, and then with new technology, with trawlers and motorized boats, we were able to go further off the reef, and we started fishing down basically what was replenishing stock for the fishery. People didn't know this, of course, they wouldn't have done it if they had, but in fishing down that replenishing stock, the fishery collapses. So it's this unanticipated nonlinear change that results in something that you thought was going to last a really long time, but it didn't. And these changes are rampant, but certainly infectious disease is by most definitions a nonlinear change, an abrupt change, sometimes with irreversible consequences. And so it's important to think about infectious disease from this perspective of increasing frequency with roots in environmental change, and how we can start to think about that. And so this is a picture that I took when I was in Uganda doing emerging infectious disease work, and we're just leaving the field site, which is in the Bwindi impenetrable forest, supposedly impenetrable because you go in there, very hard to find your way out historically. And nowadays, closely buffered by change in landscape modified for growing crops. And so you can imagine this transition zone right here is a really interesting place to do research around emerging infectious diseases. And so that's what we were doing there. And these transition zones are increasingly common. So noted, humans have drastically changed the planet, but looks like our cultivated systems, our ability to grow crops, covers about 1/3 of the Earth's surface. And there's about less than a quarter now that's left that's considered wilderness. And the wilderness we even think about in many cases, potentially like the Bolivian punishable forest, as sort of an empty forest. Like it's there, it's still a forest, but it doesn't quite have the biodiversity that it once had due to human demand. So I think it's important to consider the vast amount of change on the planet. And this IPBES group does updates regularly for policymakers, looking at the ecosystem services and sort of stacking them up and giving a status report. And it's sort of an update to the Millennium Assessment that was done. And I think this has really useful information as far as where we've been and where we're headed. And I like to think about how that affects the likelihood of emerging infectious diseases. So if we think about environmental drivers and habitat change and what that might do to populations of animals and their likelihood that they would transmit a disease to humans, I think about how those animals might have been distributed in the forest, sort of in its pristine sense. So when the forest was undisturbed, what did that look like? And we think about animals that then have to grapple with some change in their habitat. So if their habitat, if there's deforestation, say forests or timbers being cultivated, or say forests are being removed to grow palm oil or soy or some other crop, which happens very commonly pretty much on every continent, we can think about the effect that might have on wildlife species. And so this is a major area of our research right now. So to accommodate that habitat fragmentation, which I just had noted before, is one of the major reasons why mammals are listed as threatened, potentially threatened with extinction. So this is a major reason why a lot of the species on the planet are in major decline. And so we think about it sort of like this. So we have this pristine forest, maybe these are bat populations, and they're in their little subpopulation groups. So they roost together, they hang out together. They've got a very structured population, which is no surprise to those of us who work in biology. This is mostly how animals work. When you think of the red dots as a couple infected animals, and then say here there's a population that hasn't really, they're not infected. This population hasn't had that disease this year or yet. For whatever reason, these animals are all negative. And then you've started to get some landscape change in that transition zone. So this is actually a close-up view of that transition zone. And then we think about major change in that landscape. So now we've got these transition zones, and then I've obviously photoshopped this in, but this is a landscape now that's no longer suitable for those bats. So that those bats then move into the remaining isolated habitat that is available. And disease dynamics would predict that once you get populations moving in together and sharing habitat, diseases are gonna spike. They're gonna have outbreaks. The population will experience an increase in prevalence, at least for a short period of time. It's just as we all became acutely aware when COVID first came to the US, there was a big issue around travel, even within the States, because once people start traveling, they bring transmission to new areas and you create new outbreaks. And it's the same very thing for animals. And so we can expect that this is the scenario that I think warrants a lot more research. So this is where we are right now in terms of our sort of general understanding of how to think about landscape change and the emergence of a pandemic threat. So we've got forest-adapted viruses. If you go in the forest, you might get the virus if it's been adapted to transmit to humans. But if you don't go in the forest, you're not going to get it. And then over time, as the landscape has changed and the species diversity goes down, as it tends to outside of a forest setting, you've got some adaptable animals here. So these are some primates and bats. and rodents that are adaptable to this transition zone, and they're even adaptable when they get into the more urban landscape. And so the viruses that squeak through here, and I would argue this is where the diseases emerge, where they first happen. There's people and there's animals interacting in an accommodation of landscape change. The ones that make it through into urban adapted, whether they be mosquito-borne or like we were talking about the hantavirus that emerges in these more rural settings, but still human adapted. These viruses can cause regular outbreaks into people. And if they're human to human, of course, then we don't even need to think about the animal reservoir at all. But this is where we think about the risk could be highest. And so this is where we tend to do a lot of our research around emerging infectious diseases. And so I guess I wanted to mention how basically we can think about pandemics as a black swan event where we don't know what's happening. We can't imagine the conditions around which one might arise because they're so rare, they haven't occurred very commonly. But if we think about all the spillover events, all the different ways that animals have transmitted different viruses to people, we actually have a wealth of knowledge, and we can start to understand that this is a consequence of how we use the environment, how we share that ecosystem with animals, and how we either increase or decrease the opportunities for disease transmission. And so I think we're at a bit of a tipping point now in terms of what to do next. And I would say that we're very much leaning into the pandemic era. And I hope that we do. I feel like the gains, certainly in the work that you all do, our understanding of infectious disease, the spectrum of symptoms, the difference between the whole range between droplet transmission and respiratory transmission. I mean, there's been huge gains in science over the past year that wouldn't have happened if we weren't also focused on this emerging pathogen and able to pivot and study like has never happened in society before in time. And so we're starting to do that in my field as well. So we've started an EpiCenter for Emerging Infectious Disease Intelligence. We're funded by NIAID. We're one of the CREED network research centers, and we're building our surveillance with the other 10 or other 9 CREED network research centers to do emerging infectious disease surveillance in collaboration with colleagues all around the world. And so we deploy what we call a One Health surveillance strategy. So we're looking for disease X, but we're also very keen to find near neighbors of existing pathogens like coronaviruses, Ebola viruses, and of course the arboviruses. So we take into account what we understand about risk from other viruses, and then we set up surveillance by looking in cohorts where we have basically a system with health workers that let us know when there's fever in the community, when individual households are dealing with a fever, and we go in and we sample the person, the mosquitoes in and around that setting, as well as the animals. And this has really been what we call a concurrent surveillance strategy, which has been very interesting in terms of starting to reveal the transmission dynamics that are at play typically before we see outbreaks in humans. And so this work is very early on. It's really hard work to detect these viruses. They're super rare. So we have our work cut out for us for sure. But we have a lot of knowledge about which species are high risk and which are sort of the main suspects to think about in terms of wildlife. And so this has been evolving. A lot of this is building off work that we did with the USAID as part of the PREDICT project. We deployed this One Health surveillance. And really importantly, the work that we did in PREDICT with colleagues all over the world was to look at concurrent surveillance in humans, in wildlife, in domesticated species at the same time. And we deployed what were essentially One Health teams that were just okay to sample all of these. And we used techniques and sampling techniques and testing techniques that could be broad spectrum, that could basically tackle all of these different viruses that we were interested in, the ones with pandemic threat, through this sort of multivalent design where we could test for animal pathogens, human pathogens side by side in the same laboratory with the same approach. And we really think that this is where the field needs to go to be able to capture the unknown unknowns and the emerging pandemic threats. We need to have a much more closely knitted animal and human surveillance community, so that knowledge is of course shared, but techniques and tools are shared, and that one knows what the other is doing. And so that's how we rolled out the whole PREDICT project. As I was describing, we worked in some settings that were right there. Here's us sampling rodents in somebody's house, in a urban setting in Kathmandu in Nepal. We do this very safely, and we make sure that we collect samples from people and animals concurrently, so that we can think about ways that these pathogens are shared. And we also looked at bats, of course, and bats, they love to congregate. And here's a picture of bats in a tree roosting. And we can also expect that where bats are on the menu, that's a really important place to do surveillance. And bats are on the menu in a lot of places, both rural settings and urban settings, not surprisingly. And so this is an very important group of species to be tracking really closely for emerging pandemic threats. Our work is super challenging in terms of getting out to the regions. And here's a team in DRC that have done this work for decades, getting cold chain out across to where it needs to go, to the remote places where some of these animals are, and communities, rural communities that would be sampled concurrently. We also had to develop really novel ways of getting samples from primates. So here we are throwing out these juice-soaked swabs that these primates will chew on for a period of time and then spit them out, and then we go get them, and then we have a complete oral swab that we've actually validated to be able to detect pretty much any RNA or DNA virus that would be in the oral cavity. And we're optimizing that technique. And we really think that the future of disease surveillance, if we're going to truly lean into the pandemic, is through this collaborative disease detection workforce with these colleagues all over the world, many of whom were leading the charge in detection. And when COVID first emerged, they were one of the first people to detect COVID on an international spread in their own country. Even before there was a test for the new SARS-CoV, even before it was really noted or described with sequences shared, these amazing colleagues were able to use technology that we had deployed, just broad spectrum technology through the PREDICT project, to detect pathogens. And they found SARS-CoV-2 in those samples. And so if I may, and I think one thing the pandemic has really highlighted is the huge sense of humility around which we need to have in our understanding of emerging pandemic threats, and just how little we know each time one of these viruses emerges. And so I think that that's a huge call for a much broader surveillance approach, a much broader system, if you will. I call it an early warning system, but really it's a rallying cry for these colleagues and scientists around the world to be engaged and available and working on these things in between outbreaks, so that we can recognize potential emerging threats at the earliest opportunity. They're the workforce that has the talent and the know-how to do the work. And they're our safety mechanism, no matter where you live on the planet. And then it's also, like I was saying, a really unique time where we need to think about innovative technology and have some real discoveries and ways to bring this to scale, ways to sample that are more efficient, and improved ways to think about risk characterization, including bringing the pipeline to the work that you all do. And for vaccines, I mean, most importantly, we have a lot of knowledge on viruses. It's right now very hard for us to predict which one is going to cause a pandemic, if we've only found it in animals so far. And then I can't leave without mentioning the importance of multilateralism and collaboration with other countries. That's incredibly fundamental in the work that we do, and how responsible we need to be in ensuring that there's equitable sharing in benefits for all populations that engage in this work. So I'm just going to leave you with a way, and if you're interested, the PREDICT project wrapped up last year. Here's a way to find the PREDICT data. We did discover about 1,000 viruses, some of which have a pretty important fingerprint in terms of potentially being zoonotic. And so that's a resource that we have. And I did just want to acknowledge my amazing colleagues who work with me on the EpiCenter for Emerging Infectious Disease Intelligence, and the wealth of experience and talent in the PREDICT Consortium that fostered a lot of this work. Thank you so much.
And I am stepping in for Jonathan, who had technical difficulties. So thank you so much for that amazing talk. It's really just, crazy to think about how much we have to think about the environment and nature when we talk about the impact on human health. There are a lot of questions and we have very, very little time. So I'm going to try to pick and choose the ones that I think you may be able to answer in a short period of time. One of them is there a worry that broad-acting coronaviral antivirals that are being developed for this pandemic response and then possibly being overused by agri-veterinary interests could drive resistance prior to the next human pandemic? And if you can answer that in like 12 seconds or less.
Yeah, thank you. I would be much more poised to answer that question than me. I would say from the antibiotic perspective, that is a huge concern, and we can think about similar things occurring with antivirals, certainly. I think viruses are they're such much shorter lived in the environment. So I think the risk is not as big. They need to be in a host for the most part. But I think certainly driving resistance for therapeutics is a major concern, without a doubt. Is that under 12 seconds?
Yeah, that's great. We have another question. We're increasingly organized as an industry, this is from Jay, on pandemic preparedness. But beyond coronaviruses, what would be your top 3 viruses to receive disproportionate attention for anticipatory drug discovery?
So beyond coronaviruses, which I would argue is the main group, I would the paramyxovirus family, which has got Nipah virus in it, it's got Hendra virus in it. These viruses have spilled over. They have not had sustained human-to-human transmission in a major way, but we need to be on the lookout for them, as well as the Ebola viruses. We probably only understand a fraction of the Ebola virus family right now. There's a new one discovered, part of the PREDICT project a couple of years ago, and there's certainly much more to learn about that virus family and the potential, again, with all that environmental change occurring, that viruses that emerge in settings that were once rural are not rural anymore, and they're not going to stay put.
Okay, I think I have time for one more quick one because nobody's yelling at me yet. This one is, how can fever surveillance for diseases like malaria be leveraged to get better surveillance for emerging diseases as we try to improve malaria diagnostic and treatments?
Yeah, I think so. The surveillance programs that are in existence for malaria is certainly something that we think about in collaboration, especially through work that we do through USAID, because those are major surveillance systems that are set up to detect exactly that, the very non-definitive response that humans have, which is fever. So when it's malaria, that's great, treat them for malaria, but please, for gosh sakes, also check for these other pathogens that could be at play, including down the line, if nothing tests positive, an emerging pathogen, using that multivalent technique.
Yeah. Very, very good point. Okay, this has been awesome. Thank you so much for closing out the symposium. I want to pass the baton over to Jay, who is going to give us a couple of concluding remarks, and appreciate everybody's contributions and attention to our first NSofT symposium. Thank you.